yellow mosaic you can see. And this is a solanaceous crop like see potato. There was no disease in potato till 95, 96. But from 96 this kind of an apical leaf started appearing, and we characterized it and we found out that it is the same virus that is infecting tomato which has gone into potato with slight sequence differences. And tobacco we are all familiar, and chili pepper also we are all familiar with that. Ocra yellow vein mosaic virus is perhaps the oldest bigamo virus reported from India by Kulkarni in 1924. And so this is really, earlier we had only this typical yellow vein mosaic, but we have now what we call as leaf crumple and curling and enations, and like that, I will come to the number of viruses affecting the crop, the Indian scenario, will come to that. So we were thinking that the ornamental plants will be free, but this is a clerodendron, and even in your campus, no clerodendron is free of this disease. And this is Tacitus, and of course this is carrot. Okay, the legume is a favorite subject of mine because we all worked on that. And we have in black gram, in cowpea, in soybean, in dolichos, in all these legumes we have these white plate transmitted mosaic viruses. Papaya, you are familiar. And cucurbit is, I tell you, after we all started working, I mean right in front of our eyes, cucurbit's diseases became very severe. Say, starting from about 95 onwards, the bitter gourd, the cucumbers, and this time in Maharashtra, it's a big epidemic of cucumber and bitter gourd that the traders do not know how they will get the product for their sales. Tomato, of course, all of you are familiar with, chili pepper. And why I showed the weed is, the weeds have started getting attention only in the last 5 years in India, mainly because of the technology. Because weeds are very tough, robust, and it's very difficult to isolate DNA and then to do any characterization. So perhaps it was neglected, though all the while we all knew that the crop viruses must be having some relation to the weed viruses, and there must be some interlinkage. But it was very difficult to prove any part of it because the weeds were not getting characterized. But last 2 years, there is a very good attempt, like Busha's group from Madurai has reported on Vernonia, and this is from Rincosia by myself, and this Abutilon, which is the mother of all plant viruses, which occurs mainly in Brazil earlier, but now reported in other countries also, like in Indonesia and Honduras and all. This, mainly the virus spreads because this is grown as an ornamental plant for indoor decoration. In all European countries, in five-star hotels and in any seminar halls, this is kept as a decorative piece. But I happen to see lot of bushes in Ooty which was affected by this virus, and I thought that in India nobody has recorded it, and so I quickly made the characterization. It is the same Abutilon mosaic virus which occurs in Brazil. So in which case it's possible that from Brazil some Britishers, before when they were colonizing India, perhaps they might have brought it and then planted it like that. And this is the Acaripa, which is yet to be characterized. So this is a total picture. I show this colorful picture because of the problem. Like it is true that all of them are having about 60% identity in their DNA A component, and so it is very easy, to differentiate the thing on the basis of the sequence, and we can say whether it is the Asian virus or old world virus or new world virus. But definitely there is a distinct phylogenetic relationship that Indian viruses do stand out. And the most important aspect of it is this legume viruses. And this is mainly done by Professor Vestambi and my group, that the legume viruses, they have only a very basic, about 59 to 60% only sequence identity with the other bigamo viruses occurring in India, like cotton leafer, tomato leafer, bitter and all. And so genetically isolated that they are not transmitted to other hosts, and from the other hosts the viruses do not come to legumes. So the legumes represent a very, very isolated special group, and there is even a suggestion that maybe it could be raised as a separate genus. But I tell you, the sequence homology is about 60, so there is no meaning, and genome organization is more or less the same, so there is no justification to raise it as a separate genus. Okay, this is the life cycle. I wanted to do it on the board, but the slide is available. So, white flies or let us say leafhoppers, the vectors directly put the virion particles inside the cell. So, the virion particles enter the cell. This you should remember. This is a striking difference from animal viruses because there are even bacterial viruses where we are familiar with the shedding of the coat protein outside itself and then the nucleic acid coming inside. No. Here the virion particles enter into the cell and there the protein disassembly takes place. The coat protein molecules are removed and then the naked DNA comes. And this virus, for all its life cycle, it is confined to the nucleus, not in the cytoplasm like potato viruses or other cauliflower mosaic viruses which occur in cytoplasm. So in which case ssDNA, single stranded DNA that is released, should enter into nucleus. The assumption is the coat protein has got the nuclear localization signals, and so it is possible that it binds to the ssDNA and vehicle like transports it to the nucleus. And there, whatever the encapsulated single stranded DNA, we call it as virion sense DNA. Okay, that is only single stranded virion sense we call it. But this virion sense DNA will have to be replicated and it should become double stranded. So, and the double stranded molecule only is present in the most of the time of the life cycle. Okay, but the very funny part is that nobody knows how this is done. We know that this virion sense DNA enters into nucleus and this DNA is copied into the double strand, probably using RNA primer. It is not certain and it is probably using an RNA primer. The double strand is made, but the job is done by the host DNA polymerase. And from where it starts, this is only an assumption and this is only a very, very big assumption that it starts from here. So we are talking about the virion sense, that is plus sense, from which the minus sense will have to be made, and the gap, there is, why I am telling about the gap is, if you write a project, you will get it. Nobody knows this negative sense from where it originates, what is the primer and who does the job. Okay? So it becomes double stranded DNA. Let us say the gaps are there, but let us say the complementary sense is done and the double stranded is done. And it is the double stranded DNA which is the active molecule both for transcription and also for replication. So from that the virion proteins will be produced. I will tell you how it is done. And the one of the protein is what we call as the replication initiation protein, and that will only cause the nicking, the splitting of a 9 nucleotide sequence. It just opens up so that the 3' OH end is created, and it is the host DNA polymerase again copies the circuit. So the method by which it replicates is called as rolling circle replication. People who are familiar with the bacteria, we know that it is on a circular template and one template remains as such, and then the molecule goes round and round, round and round, round and round, and then it comes out like that. And that we will see shortly. So some of the circles will enter into the transcription mode and will produce the host proteins. Some of them will enter into replication, and if the proteins are already made and they are available, they are encapsulated and they may come out as protein encapsulated molecule, or the movement protein that is translated will bind to this double stranded DNA and it will move from cell to cell. So this is the overall, this picture, if you remember, you have understood Gemini. Okay? This is to tell you the details. I think many of you are familiar with it, but I can quickly go through it. So in the case of bipartite begomoviruses, I told you that DNA A is having the proteins responsible for replication and code protein. So this is the replication initiation protein, this left hand side molecule, and this is the code protein. And here it is jam packed. There is no non-coding region at all in this virus, and we get a small non-coding region only in Mercury virus. And between A and B, there is a small non-coding region which is near identical, and those sequences have to be identical so that the red protein binds and identifies the thier of the B also, so that the B is also replicated by the same replication initiation protein. So the B is dependent on A for its replication and encapsulation, because the same coprotein molecule only surrounds the DNA B molecule also. So both encapsulation and replication, it is dependent on A. And A is dependent on B for movement. This particular protein is called nuclear shuttle protein, and this is responsible for taking the double-stranded DNA inside the nucleus and also to take it outside the nucleus, both the nuclear import and export. And this is the one which takes it, the complex of the nuclear shuttle protein and DNA, to the periphery of the cell, and from the cell it docks it on to the plasmodisk matter. So this does the job of cell-to-cell transport, and this does the job of nuclear transport, and this has got the same common region where the promoters for this ORF are present and the sequences which the replication initiation protein identifies and binds. All these sequences are present here. You can stop me if you do not follow. If the time permits, that's okay. The questions are permitted later.
Yeah.
I mean, if it is, because I've seen once I lose all of you, then I go on rattling away and nobody's game. That should not happen. And so, why these circles are shown, you may be wondering. Like, there are lots of circles involved now. In the last 10 years, the understanding on the characterization has improved, and mainly the situation is very bad in India. Okay, we know that AMB exists, and one example is telomerase virus of Legionella. But the monopartite genome is present even among bigomoviruses, where A alone can cause very good symptoms. That is one group of virus. Example is tomato-leafal Karnataka virus, where A alone can cause very good symptoms. But in most of the cases, the monopartite DNA A alone is not able to cause the symptoms, and it requires a satellite DNA, like in the case of tomato-leafal Bangalore virus, and that is a combination of monopartite plus beta satellite. But the typical, another example is the cotton leafal viruses complex, where you have a DNA A, you have a beta satellite, and you have an alpha satellite, which is just like the red protein subclass of nanovirus. Earlier it was called as DNA 1, but now they have changed the name to alpha satellite. Yes, sir.
Whether these beta satellites can be viewed under EM?
No, they are, okay. He has asked me a very good question. He asked whether the beta satellites can be viewed in the electron microscopy. It's a very good question. Because how these beta satellites are encapsulated is not yet understood. One theory floated by the German group is that because the DNA size only determines the particle size, what they say is, when you are seeing in the electron microscopy, you will see lots of one isometric particles. Not a paired particle, but a single isometric particles you will see in many cases. And when they did the DNA isolation from that and isolated it, they found that many of them were subgenomic DNA of about 1.3 kb. Inside the plant, when the DNA replicates, maybe polymerase sometimes it slips or it doesn't move the entire circle, in which case it generates lot of subgenomic molecules which have undergone deletions, and these are very common molecules in any naturally infected plant. So the hypothesis is that because the subgenomic DNA are encapsulated in these kind of single particles, it is possible that the beta satellites are encapsulated in these kind of single particles, but the final proof is not yet given. Okay, and by, of course, biochemical studies, like you can do the acrylamide electrophoresis, isolating the particles, and do both DNA PCR and your coprotein antibody test. By that, it has been proved that yes, beta satellite is encapsulated by the same coprotein molecule. But whether the isolation, the isolated particles or the paired particles is not clear. Sometimes I feel even in the paired particle, the DNA, the one DNA 2.7 kb can be there, and along with that within 1.3 kb, the size restriction is there. It cannot take in more than that. So it should be either 1.3 or 2.7.
Sequence similarity is there.
Pardon?
Sequence similarity between that DNA A and the.
Okay, I was coming to that. Like DNA A and beta satellite, except for the 9 nucleotide sequence which represents the DNA cleavage sequence or the origin of replication, there is no sequence similarity between DNA A and the beta satellite. But if you are first time characterizing, you will get lot of circles, and if only money is not a crunch and we can get all the 100 molecules get sequenced, my belief is, because it has been shown by Keith Saunders in Ageratum, that there may be some circles like my student here Neha got like that in Pune samples of tomato. We will get certain circles where the half of it will be DNA A and little bit of it will be beta satellite. Like that, I feel the beta satellite must be derived between the host genomic DNA and the DNA A, but the derivatives, how many of it are having the sequences of A and sequences of host, and finally all of them getting eliminated, and we get only the functionally very efficient beta satellites, which retains only this 9 nucleotide sequence which is common to the origin of replication of the DNA. Earlier days we were doing isolation of the DNA and then cloning it by the cesium chloride method. These are the representative of the open circular replicative forms. In southern you can see this, and then pick up the fraction which is very good in the replicative form, and then cloning it. But always the problem is the virus concentration is very low, and most of the plants from our tropical conditions it is very difficult to deal with in isolation of DNA. And then we have to deal with 1.3, 2.7, and then we have to get A, we have to get B, we have to get beta satellite and alpha satellite. It's very, very difficult. And in most of the cases it is mixed infection. So we are going to have 2 or 3 viruses present with 2 or 3 components in it. And again the variability. We cannot have an identified primer by which you can clone all the genomic components. So when we were all struggling like this, then there came this technology of pre-29 DNA polymerase, which is very, very efficient on the circular template, and it also has got the 3 to 5 exonuclease activity, which is the proofreading activity. So this is in summary of the rolling circle application. We can get lots and lots of DNA molecules generated. Like I will show you some of the slides like this. So here you isolate the DNA, and you require only some 50 nanogram of it, and you have a random hexamer, so that the DNA, and use that for bacteriophage P29 DNA polymerase. The initiation of the circle starts at different points, so that you have got lots of double-stranded DNA, what we say as concatenous. And once you concatenous is like a chain of 2 plus 4 plus 8 plus 16 plus 32 plus 64 plus like that, it's all like a huge mass of DNA. And if you use any restriction enzyme, and luckily if that enzyme has got unique site, means only one site, then chippy your concatenous are cut into single 2.7 kb genomic components. And I tell you, it has made our life. I have shown in different ways how the circles can be generated, and that is called the rolling circle amplification procedure, which has been very much popularized by Jesky, but before that there have been 1 or 2 papers who are working on other circle ways. So once you have that RCA product, you can go for cloning, and it is so rich that you can even directly go for inoculation, or you can go directly even for sequencing, and that makes your life very, very easy. And now onwards we used that method only. So this is how the rolling circle DNA looks, like a lot of smear, and if you cut it, only thing is your luck plays a role in this. If you are very, very lucky, you will get the 2.7 kb linearized DNA A, 2.7 kb linearized DNA B, and 1.3 kb linearized beta satellite. Everything you will get it in one go, but it all depends on your luck. Sometimes you won't get it. And then you will do your usual cloning and then restriction. classification and find out well how it is and all.
DNA A and DNA B will be of same size, not different.
Say exactly, yeah, you are very correct. DNA A and DNA B are same size. And earlier Goodman and all had done an alkaline sodium sequence radius centrifugation where they try to separate the A and B. But now nobody does it. You just take 10 clones and then send it for sequencing. Because the sequences will tell you which are the clones B and which are the clone A. The advantage of A clone is that across the world, white-plate transmitted bigomoviruses, they have minimum 60% sequence identity. So if you have A clone in your hand, it could be a very peculiar clone, but even then it is hit with some DNA A of some island. Whereas the difficulty comes with B, because you won't have any, B the homology is only about 42 to 45%. Whether it is within Asia, within India, within old world or new world, the homology is only that much. So B, you can only, if suppose, I always say, if a clone is not hitting with anything, most probably it could be B. In which case you go for translated blast. When you go for translated blast, the movement protein has got typical signatures. So the movement protein amino acids will be picked up in your blast analysis, and at least you can have a hope that it could be a DNA. Otherwise you cannot differentiate. So since I thought Professor Belithavi would have covered a lot of yellow-mosaic viruses, I took up tomato leaf curl, which is another field in which I have worked quite a lot. So right today, by doing this rolling circle only, quickly we finished in 2 years all these viruses, and we know now that about 16 viruses are infecting tomato leaf curl, and we have the infectious clones of these viruses in the respective groups that I have given, and we do have the 2 bipartite viruses. One is the biggest enemy, tomato leaf curl New Delhi virus, and another is the tomato leaf curl Palampur virus. It's very important because Palampur is in Himalaya, and so that means earlier, we were thinking in colder climatic conditions you will not get these white-plate transmitted viruses, but of late we are having potato and this Palampur virus. This picture is a very colorful one, and that is why I showed you. This only shows wherever Maharashtra, wherever the cultivation of the crop is very, very intense, you do have the number of viruses more. And the tragedy is, as I will be telling you, that each virus may have another one set of beta satellites. Say for example, tomato leaf curl Bangalore virus, when Neha took the sample from Bangalore, it was Bangalore beta satellite. Whereas when she took the sample from Maharashtra, it was another beta satellite, which is called as Thailand tomato leaf curl beta satellite. So this kind of a complexity doesn't occur in anywhere in the world. We have one virus DNA A which can promiscuously combine with many beta satellites. And depending upon that, the host range is going to vary and severity is going to vary. This is all only tomato leaf curl New Delhi virus. I only wanted to show you the relationship of all the isolates of tomato leaf curl New Delhi virus, and this is the one which is infecting tomato, which is having highest sequence identity. But tomato leaf curl New Delhi virus is reported on all the other things will share between 90 to 92. In the case of bigomovirus, because that question often comes, I will come to that. Once you have got A and B components settled, and A you have sequenced, the taxonomy of the virus group is dependent on the DNA A complete nucleotide sequence. Okay? Supposing you have cloned it, and if your sequence is hitting and having identity of 89% with the other DNA A, then you have to call your virus, but that virus only. Say for example, when you are doing the blast analysis, it is hitting with my isolate of tomato leaf curl New Delhi. Then you will call your virus by tomato leaf curl New Delhi only, though you would have isolated it from Coimbatore. But if your sequence is even less than 89, that is even if it is 88.9, then yours is a new species. So that is the species demarcation threshold, and that is how all these virus names have been tried. And of course, if you are putting the city name, country name, then it is slightly helpful for us from where the virus has been isolated and characterized. But you may be asking why at all we should characterize. Basically, we are interested in increasing the productivity and reducing the disease. And any breeding resistance can be improved only when your inoculation protocol is very, very good and reliable. All the people, pathologist people are asked to do the screening by the breeders, and the whitefly transmission is the normal method, which is very, very difficult, maybe because we are handling a very peculiar genotype of whitefly, or the environmental conditions are not proper, the glasshouse conditions are not proper, and we struggle, and by the time the time is over. So if only you clone and then you make these constructs in Bin19 based vector or TA plasmid vector, and then you mobilize it into agrobacterium, the agrobacterium can be, having the construct can be used as an inoculative things as I have shown here. This is the common laboratory host Inoculation of Balsamiana, and these are the tomato, and this is how we do the inoculation. So once you have cloned, I know many people do not agree with me because this is a very severe way of testing the plant. But if you are going to have a marker for the resistant gene, and it should be an ultimate test for resistance, so I believe that agroinoculation should be used, or grafting, where you are sure that the viruses are getting introduced into the plant. So this is how we have done inoculation in tomato. And say for example, this LA1777 is the only one which has got the resistance against the tomato leaf mildew virus. And whereas the other variety, this one H24, was earlier very famous because it was having the TY1 and TY2 gene combination, and it was supposed to be resistant, but now it is not having any resistance. See, this is the way it can be done. Like we have got this cloned component to every individual genotype having different tomato yellow leaf curl gene, that's why it's called a TY, but we do not have tomato yellow leaf curl virus. But the gene is used in the genotypes, and so they continue to use the term, the breeders use the term TY1, TY2, TY3, TY4 very commonly. So the combination of Y1 and Y1 also is susceptible. Y search to a very high level if only you have these agro-constructs with you. That is the only advantage. When you are using whitefly and when you are using inoculum source, you do not know what is the virus you are handling, and you do not know whether your whitefly does the job. So this is the lines in the trial, and you can see this is the susceptible line and this is the pyramidal line. Maybe in another 1 or 2 years this is ready for use. So this is coming to beta satellite. It has got only one open reading frame, which is beta C1, and it is supposed to be the RNAi suppressor. Means if the RNAi defense of the plant is triggered, this suppressor molecule can suppress the defense reaction. And the the mechanism by which this beta C1 does also is slightly there is a indication. Like in all these RNAi defense mechanisms, the protein is degraded by ubiquitin proteasome degradation method. So this beta C1 binds to the ubiquitin ligase. This has got one very big replication initiation protein. Other than that, it does not have any other protein. And we can get the disease symptom expression by beta satellite alone. And only in 1 or 2 cases they have shown that alpha satellites may be interfering with both DNA B and beta satellites and reduce the symptom expression. Attenuation of the symptoms could be one of the role of the alpha satellite, but it is not at known. So beta satellites, the headache is that only we have, especially this is a hot spot, the Indian subcontinent and Southeast Asia are the things which have got diverse beta satellites. So the beta satellites, the beta C1 protein is so different that we have got hundreds of 200s of beta satellites now. But they all have this region, what is called a satellite conserved region, where the sequence is identical across all the beta satellites. But the beta C1 is very diverse. There is no protein signature you can see. We cannot predict any function for it, and it is such a unique protein like that. So what is the beta satellite role? when we checked it, we found that the beta satellite, when you are inoculating, it is increasing the severity of the symptoms. In fact, in many of the viruses, when you are inoculating DNA A alone, you won't get any symptoms, and only when you are inoculating along with the beta satellite, you will get the symptoms. The second thing that it does is that the DNA level of A is, the concentration is more when you are inoculating with beta satellite. You can see that this is A alone and this is AB, whereas the A plus B plus beta alone, A plus B plus beta way, you are getting more beta satellite concentration. This also we proved it by real-time PCR, where the A concentration and B concentration are more when you are inoculating with the beta satellite. And one interesting finding is that in South, many of these tomato varieties are released by Agricultural University Bangalore, and they are considered to be very resistant to tomato leaf cut Bangalore virus. But when we inoculated with the beta satellites, different beta satellites, there is no resistance to it, and that is the biggest problem. Say for example, tomato leaf cut New Delhi virus is associated with one set of beta satellite when it is inoculating tomato, but when it is present in cucurbit, it is present with another beta satellite. And whitefly must be transmitting across from this host to that host to this host. But then the beta satellites, though they may get transmitted, they may not get replicated, and they may get suppressed in another host. All these are nothing understood. And mixed infection is the most common problem in India. And that is why, we can never be sure what are the viruses present in the sample. Even you people, if you have taken samples for your work, 2 to 3 viruses will be definitely present in it. The mixed infection with Palampur virus, we find it very high in northern India, and we could see that the A and B of these 2 viruses can be swapped, and that is what is called as pseudo recombination. Like we inoculated the New Delhi A with the New Delhi Palampur B and Palampur A with the New Delhi B, and we found that the symptoms are very different when you are swapping the fragments, and all of them, when they are present together, the severe infection results. And not only that, whitefly could pick up all these virus components and transmit the disease complex, that is A, B, everything is complex into the thing. This is for the Indian scenario, never make a mistake that Bindi is having only Bindi yellow vein mosaic. Right now we are having 13 viruses, and by the time I complete my talk, it could be 16 viruses. The same, another example is tomato. These are the acronyms like Bindi yellow vein Delhi virus, Bindi yellow vein Haryana, Bindi yellow vein Bhubaneswar. Like that, this work is from Dr. Krishna Reddy from IHR. And tomato, of course, all over India, like Suprayo, myself, everybody had contributed to it. So tomato, you see, is affected by more than 30 viruses. So now, what we are talking is not a single virus, and unless you know that, you cannot breed for research. And what you breed here in tomato, in TNAU, will never hold good in Karnataka. So, this kind of a complication we are having it. I have listed it, and this list, any one of you can take it, though it is unpublished. It is only the computer work, so it's nothing original. Say for example, this papaya. Papaya still is not clear. There are 4 viruses, but I feel it should be more. The same way chili also. Chili is there? Yes. Chili also you can see, the viruses are more. And so, here I am concentrating only on DNA A, and we are not sure this A is going to combine with how many beta satellites. Then the combinations are going to be infinite, and that is the exact problem against diseases in vegetables I wanted to put across. This is only to sum up, because there is a lot of confusion regarding whitefly, so I thought this platform I will make it sufficient to clarify certain things. Whitefly is the vector, and this is very easy to imagine, and many of you are very familiar. The virion particles are entering it, and they go through the intergut, foregut, and then there is a midgut, and then from midgut it comes into the body cavity, what we call as hemocoel. And from hemocoel it should find its way into the salivary gland, and so the next time when it is feeding through the salivary gland, it will enter into the plant. So it has to go through, this is the unipolar or one-way traffic. It goes through up to midgut, and from the midgut it enters into the cavity, and from cavity it has to go to the salivary gland. About 6 to 8 hours it takes for the translocation of these virion particles. And white flies are, there are two origins of center of origins for white flies. There is a strong belief that Africa is the primary origin, center of origin, but Indian subcontinent is also one of the center of origin of white flies. So whenever it is a center of origin, then the diversity, everything will be very high. So it is possible that white flies that we are having belong to different types. There was a term used as I skipped this. There was a term used to refer to these vectors as biotypes. That is, they don't differ in their morphometric characters. You cannot distinguish them, but they differ only in their certain biological characters like transmission of the virus, insecticide resistance, and the preference to certain hosts. These are the biological characters. So based on that, the term biotype was used by Costa. And it so happened that in USA, they had severe diseases in California, in Rift Valley and all. And there, when the disease became very severe and when they looked into it, they got the white fly which belongs to the Middle East. But they coined the term to differentiate the normal American white fly as A, and the one which they present in actively transmitting the disease, they called it as B. And so the B white fly term was used very much in Nature and Science. There were a lot of articles on that. So once these two terms A and B were used, what happened was the rest of all the laboratory in all the world, they characterized the mitochondrial cytochrome oxidase subunit gene, and they called it by the same alphabetical, up to Z it went. But Gibaro in last 2 years he has been working, but the Annual Review of Entomology, the article came in 2011, he thought enough is enough. There is no such nomenclature should be given, and he had given beautifully this definition. See, these various white fly populations, they have been differentiated mainly based on either ITS region sequence or estrase pattern, or based on cytochrome oxidase gene sequence. Nobody has considered the biological character very much into the thing for classification. So why you people are calling it as biotype? It's wrong. So he did thorough analysis and he said that these are all genotypes, and based on the mitochondrial cytochrome oxidase gene, he recognized about 11 major groups which are blue, and the individual which he calls it as putative species or genotype. And based on that, please do not use the word biotype. Even if your professors and all are telling you, please tell them that no more the biotype term is valid. The term used is either putative species or genotype is the term used. And so we wanted to know, because we were working on legume viruses, and the legume viruses are not transmissible to the other host. We had a funny hypothesis that it is possible some very different type of biotype may be involved in the transmission of these viruses. And so we collected all over samples from India, and we found that the genotype occurring in soybean is Asia27, Asia21, which is not yet reported from India. Ours is the first study on that, and Asia1. And Professor Muniappa group had reported Asia25 and Asia28, and this is that dreadful B biotype, which is now called as MEAM1, which is nothing but Middle East Asia -1. This is the biotype B. See, you are always tempted that when somebody reports something abroad, we also try to see whether that B is there at all or not. All of us have wasted our time like that. But what we have is a better transmitting agent, Asia21, because this causes the very severe disease of cotton leaf disease transmission in Punjab region in Pakistan. So now we have to look for wherever such big epidemics of severe disease are coming up, whether it is due to these kind of peculiar populations of white line, with that we should see. And from one genotype to another genotype, the sequence diversity is just 3.5. 3.5 is a sequence diversity based on which the putative species are differentiated. So I can sum up. I told you that the virus diversity is there and the LDV is there, devastating pathogen. And it is not that only your B biotype is a transmitting active transmitter. No, that kind of a misconception please remove from your idea. See, when Americans or Europeans or somebody talks like that, because they have not seen what is the disease in that kind of an epidemic proportion as we all experience in India. Okay, so another big thing biotype that is talked about is Q biotype in USA. So the B and Q are supposed to be the one replacing each other and all that stuff. But I tell you, Q biotype was very much existing in all northwestern India where cotton is growing. So all these are now very much clarified. The earlier B biotype is NEAM1, which definitely occurs in Karnataka and to some extent occurs in Tamil Nadu, Andhra Pradesh and Gujarat, a pocket. But the rest of India, it does not occur, especially northern India and central India, it does not occur. And we should be more bothered about the other active transmitters, because the peculiarity of Asia2-1 is it's highly resistant to many of the common insecticides that are used in the cultivation. So these are the problems that we should be familiar with. Well, I always advocate for agroconstructs for resistance breeding, and the marker is one big thing that we are all chasing, but I am very sad that Germany is not here. Like for example, yellow mosaic disease, even moonbeam and black gram markers, none of the markers work really for any resistance breeding. So maybe some Illumina sequencing we may have to do to quickly arrive at some of the markers and microsatellite data. And first fix up the baseline data. We do not have how many viruses are infecting tomato, papaya, any crop for the matter. And we should fix up a target. 5 years from today you take it and you just tell me how many viruses are infecting. Then only we will know which virus is coming newly. We have no idea. And so that is the first submission to start with. So this is our team, and Neha and Surya are here. He worked on Palampur virus, Surya is here, and Neha worked on Bangalore virus. And in fact, he is my uncle, but he does the agro inoculation for all of us. And that's why I brought him over here. And this is the Costa's very famous quotation. Yes, when one of the British Council scientists visited India, he was surprised to find that all your roads are full of infections. You mean to say government, there is no rule to remove these things? Then I realized that your backyard is full of viruses. So at least we should not work on anything else, but should work only on viral diseases, I feel, because very, very big threat to the cultivation of the crop. And of course I acknowledge the funding from ICR and the BBT and IRI and the CSR Emeritus Scientist team in which I am working. So thank you very much for your patience.
We cannot just say that we have a sequence. Only the virus. So they say you have a sequence, and this sequence is less than 89%, and you get the virus. Another one consistency has to have other characters adding to for some serology or best biology. How is the species defined? The species is defined that the parent of this species, two members of the species, can make form an offspring. So the Vidya species, which you rightly said, are species because they cannot make offspring. So AM1, Asia1 cannot speak. In biology, that's not easy. In the virology, which is the most complicated biology, it's almost impossible. So how how do you define a biological entity of species? If we can take A, and this A will replicate a B of another virus, and then A replicate B of another virus is the same species. You have now transreplicated two species.
Yes. But
You need to have either tell plate of the plate, or you remove the negative.
so there are examples. What Stefan is telling about is that what is the basic criteria to tell that this and this are the same species? One criteria he now discussed is that if one A is capable of transmitting the other B, then they too should be the strains of the same virus or same species. But there are instances, especially bean dog virus and tomatochlorotic model. These two viruses, they do not have any sequence identity between themselves. But in the common region, some blocks of rep binding isolon sequences are same. So because of that, the bean dog can replicate tomatochlorotic model. But it doesn't mean that these two are one and the same virus. So as he rightly said, bigomovirus taxonomy is more complicated and I hate, say for example, you go on calculating, it happens sometimes that my computer may be stupid and it might have left certain steps. So 91 or 92% if I get it, then I will call my virus as the same virus as the others. But one fine day one entry will come and then when you resequence analyze everything, you will have the 88.9. So we hope that it is a new species. Yes, it's a very stupid criterion, this sequence identity, like vector transmission, host range, and all that should be included. But now in this present era of genomics, like I happen to be one of the members of the Genomics taxonomy, and now it is raised from 89 to 91. The latest, that 10th report is 10. It will be 91% sequence identity on the basis of which it will be separated into species. But it is stupid, I agree.
Now the question arises to me: if you have freedom, and they say we need a solution, and you give a talk, and you say, okay, monkey, yellow mosaic virus, all of a sudden we come up at the end of our talk, we add up 100 viruses and the reader says, so now.
That's right.
Okay, we need to